So today we're taking a sample from a local stream that is actually connected to Lake Michigan, which is one of the Great Lakes. And what we end up finding is pretty interesting. We find a member of the Coleps genus, which is a ciliate, from the Prostomatea class. This that we're showing right here is a little bit of a zoomed out view, just showing its movement. I believe that this specific species is the Coleps hirtus, discovered by the German zoologist Nietzsche in 1827. Now, this genus and this species are noted for the barrel-shaped bodies, and there is a regular arrangement of what are called ectoplasmic plates. The plates are biomineralized calcium carbonate, and they are basically used as a form of armor to form a hard shell around the organism. Here you can kind of see the bumpy surface, and those are the actual plates that are biomineralized into calcium carbonate. The biomineralized test that is created is both inorganic and organic in nature, and forms a mesh-like structure around the organism. Coleps hirtus is generally 40 to 65 micrometers long, and it has 3 posterior spines, as well as cilia on the anterior end. The other species, Coleps elongatus, originally discovered by Ehrenberg in 1830, has a more cylindrical cell body as opposed to the barrel-shaped body of Coleps hirtus, and tends to be longer, hence the species name, and generally observed to be 65 to 80 micrometers long. In our specific sample, we're able to see that this looks most like Coleps hirtus, but that this other example here has a very noticeable divide in the middle of the body, which you may refer to as the equatorial cleft. This species, shown a little zoomed in on the right here, is noticeably different than the one on the left, our first species. I'm not sure which species the one we're currently looking at, the one on the right, actually is, because of that. But nonetheless, it's definitely a Coleps of some sort, and also has those. I believe there's 3 posterior spines. It does appear to be slightly longer and slightly more cylindrical shaped than the first example that we saw. Coleps are known to be engaged in predatory behavior against bacteria, algae, flagellates, and ciliates as well. Coleps generally use toxicysts, which are basically a way for the organism to deliver a poison to capture the prey that it's going after. The toxicist is a form of offensive extrazoom that is basically used to paralyze the prey. In the case of Coleps hirtus, the toxicist discharge has been chemically analyzed using chromatography and mass spectrometry, and basically was discovered that the toxicist has over 19 saturated, monounsaturated, and polyunsaturated fatty acids, along with the smaller amount of phytanic acid. While they're not visible here in this magnification, because I don't have that powerful of a setup, the toxicists in the Coleps hirtus appear to be rod-shaped or cylindrical organelles in the oral basket. So they're clustered by the oral basket. This mechanism, the presence of free fatty acids or FFAs, is present in other species not related to ciliated protists, such as microalgae. For example, in the case of fibrocapsa. While it's not exactly clear as to the source of phytanic acid or its specific role in the toxicity of the ciliate, it has been shown that the production of phytanic acid can occur due to the biodegradation of the chlorophyll side chain. Regarding the potential toxicity of phytanic acid itself, it's not necessarily known as to the mechanism that it might be involved in, though there is proton translocation action that has been demonstrated with the effect of phytanic acid, where mitochondrial respiration can be impaired. So it's possible that this weapon that the ciliate uses, that the Coleps uses, has some kind of mechanism through that. Anyways, I hope you found this interesting, and thank you for watching.